Hello, everybody. How's it going? Thank you all so much for joining us again today for another live presentation. This is one that I know I say this every time that I'm particularly excited about it, but this one is especially different because we are going to be taking a deep dive into some of the really cool science happening, not necessarily behind the scenes at OCEARCH, but right there for everybody to see. We have three incredible collaborating scientists joining us today. We have Lisa Crawford from Stony Brook University. We have Dr. Harley Newton from Wildlife Conservation Society. And we have Lisa Hoops from the Georgia Aquarium. These are our researchers who they have their own projects going, but then they also are working together to really help develop our most advanced understanding yet of white shark health, and they're going to tell us all about why this is so important. I know that they say you learn something new every day. I think we're about to learn more than one new thing today, because these are some pretty groundbreaking projects that these guys are working on. So I'm really excited to turn it over to the three of you. Thank you all so much for being here. To all those people watching, if you guys have any questions, these are the people to ask. They will let you know, or they'll have an answer for you or give you the best answer they can. So please use the comment section wherever you are to leave those questions. Use the share button to spread it to anybody you think might be a shark enthusiast, because these are some shark experts. And with that, I think I'm going to turn it over to the three of you. Lisa, Harley. Don't all jump at once now.
No, it's great. All right. So I'm just going to kind of get stuff started. So yeah, the topic of today that we chose, because we all work together kind of towards the same mission, is white shark health and conservation medicine. And conservation medicine is really just an interdisciplinary field that looks at the relationships between human health, animal health, and environmental health or ecosystem health. And it really recognizes that animals are really critical sentinels of environmental disturbance. We know that shark populations are in decline, and they're threatened globally due to overexploitation from fisheries and also habitat destruction. Those are their two major issues. We really don't know currently what the role emerging diseases may play in those declines. We also really need to get a better understanding of how acute and chronic stresses, either from environmental change, or from human stressors, human interactions, play in the role that emerging diseases might have, or just on the fitness of the population. It's really important that we understand those interactions, because we need to develop health management practices for our populations, policies and programs that can sustain biodiversity for the future, and also protect ecosystems that are essential to animals and humans. So we can go to the next slide. So in order to sort of start us all at the same place, we really need to have a definition of health. And this is kind of the standard definition of health for any living being, which is just a state of physical and psychological well-being and of productivity, including reproduction. Simply put, it really is the ability to kind of adapt and self-manage. So it's the ability to take in food and stay in good nutritional condition. It's about maintaining a good body weight. It's about having normal behaviors, and it's about being in a normal reproductive status. So it's really just kind of a baseline sort of homeostasis for that animal. Next. As we go through this, we have interest both in individual animal health as well as population level health. Individual animal health is a real challenge, particularly in marine species, because physical and behavioral changes are typically not apparent. Obviously, they live in an aquatic environment. It's really hard to see these animals and observe them to know that they're behaving correctly. We do get a good look at them on the lifts, so we can get a sense of their physical well-being, but some of those other things that we consider when we're looking at health are hard to see. Additionally, health influences the way that these animals respond to handling and capture. So it's really important as we are interacting with an animal that we start assessing health early on, how that animal looks, so that we decide whether we're going to handle the animal or whether we're going to let it go without handling it. Population health is where we start figuring out what the physiological effects of environmental and anthropogenic stressors are, and also looking at the effect that underlying diseases have on population dynamics. And that's where, you know, looking at population health really is going to give us a sense of sort of the future of our oceans, and gives us a sense of how human interactions in the environment are interacting with at a species level. Next slide. So one of the reasons that we all wanted to talk together on the same talk rather than us giving talks separately is because all the work that we do is just heavily interactive. So my specialty is veterinary medicine or animal health. That's heavily influenced by the diet or the nutritional status of the animal, Lisa Hoops investigates. And then it's also heavily influenced by what sort of plastics or other chemical contaminants or other toxins that animals might be exposed to in the environment. So really, it's us together working on this project that starts creating that picture of health for white sharks. Next.
Awesome. Well, we all know that white sharks are very important for healthy oceans. All sharks are important for healthy oceans. And that's because white sharks have a top-down influence on the ecosystems that they inhabit, and they have great influence over the food web. So if you remove a top predator, specifically the white shark in this instance, you really have a cascade of negative effects that ripple through the entire ecosystem. So what happens is their prey tends to increase in population, and what those particular prey, and in this instance it's seals, what they feed on tends to decrease in abundance. And so what you get is a really disproportionate looking food web instead of that great diversity that we had at the beginning of this diagram. So white sharks are really important to maintain in systems because they create healthy oceans. So white sharks equal healthy oceans. And that's a great reason for us to continue to study them and understand the role that they play in these systems.
So that brings us to why we're studying what we're studying. So the problem we have right now as researchers is that we have a lack of baseline information about white shark health. We don't yet know what a healthy animal really looks like, but we can start to sample lots of animals and get an idea of what a healthy animal might look like from its blood or other tissues. And then another problem we have is that we have a limited understanding of how these environmental or human-derived stressors are actually impacting the animals on a cellular, physiological, individual level. We know that these white sharks are exposed to stressors, but we don't yet understand what's happening in their bodies exactly, but we're starting to work on that. And so some of the broader goals of our 3 projects and the projects we're working on together are to really understand the health status of sharks. What are the baseline health information we can get, and start to investigate the actual effects of these stressors on sharks themselves. And finally, one of our really broad goals that we're reaching for is to identify shark populations that might be at extreme risk of some of these environmental or human-derived stressors that would require increased protection or surveillance. And so that brings us to where our 3 disciplines intersect. We have Lisa Hoops studying nutrition, Harley Newton studying veterinary health, and I'm studying toxicology. And as we've kind of alluded to, these 3 disciplines intersect to give us a larger picture of white shark health and shark health in general. And so the rest of this live stream, we're going to go over each of our individual projects and disciplines, and then we'll bring it all together and talk about how this information intersects so we can start to answer some of our big questions and accomplish our goals.
So I'm just going to start just by talking about blood because that's my main focus in the primary sample that I collect. It's also one of the 2 primary samples that the 2 Lisas also need for their studies. but they also use some other tissue samples as well. You know, simply put, blood is made of cells and plasma. And in characterizing the cells that are present and the numbers, we get a sense of how healthy an animal is. And then the components of the plasma are important to study to get a sense of health, nutritional biomarkers, as well as potential toxicologic biomarkers, which we'll discuss. So the main role of blood is it carries important blood gases like oxygen and carbon dioxide. It carries important metabolites from our kidneys and liver in order for us to be able to excrete it. It carries important nutrients to parts of the body so that we can be well nourished. It does carry important hormones, which I also want to mention, you know, reproductive fitness is also a really important marker of health. And although we're not talking about it today, it is one of the major studies in the health of white sharks that we're investigating. Immune defenses are also carried by the blood. And that's one of the things that I in particular am looking at, sort of ways to detect that there's some sort of immune response in the animals. And then just basic clotting, you know, just sort of basic acute wound repair. So the photomicrograph you have on the right is an actual sample of what white shark blood looks like. And this is honestly, for me, the shark that started it all. This is the very first white shark I ever saw. It's the first shark in my study, and this is Montauk, which was a young of the year that we caught in 2016. You can see some interesting things about white shark blood, which is that the red blood cells have a nucleus, which is not the case in mammals and other species. And then the white blood cells, which we'll discuss later, also look very different. So we can continue on to the next slide. So this slide just discusses the basic veterinary health panel that we carry out on all the sharks. This health panel is pretty much no different from what your dog or cat gets when it goes to the vet. It's also pretty much no different from what you yourself get when you go in for a physical or an exam when they're trying to figure out what might be making you ill. So the types of things that we do is we do a hematocrit control solid.
Harley, sorry to interrupt. I didn't want to interrupt, but I did want to make sure that we filled in one important piece of the puzzle, because you're gonna run people through the things that we get, but it's also important to let people know you're the one actually collecting this sample. So, you know, this is what you're about to go through is one of the things, right, that you're on the lift, you get the sample, and then it goes back to the lab for all of that, just so that everyone is aware. Like, that is what's happening here. Am I right? Did I miss a step?
No, absolutely. Yeah. I mean, the blood sample that's collected on the lift, there's a team of us that can collect that. I will say that both Lisa Hoops and Lisa Crawford are incredibly skilled at getting good blood samples. And then we also have a big veterinary team that also helps. But yes, this is one of those samples that is most prominently seen being collected on the lift and then run back to the lab so that we can process it on the ship. And some of these tests we do on the ship right then and there, so we get some early information, and then other stuff happens afterwards that we send it to specific diagnostic labs.
Okay, yeah, sorry, I didn't mean to interrupt. I think that's an important part of the story that I just wanted people to know. So
no worries. So yeah, the hematocrit and total solids, that's actually something that we do carry out on the ship. It gives us a basic proportion of how many red cells are present and what the protein levels are in the blood. So it does give us a quick assessment of health in the animals, and it does vary by species and lifestyle. So it's something important to look at. A complete blood count is something that we carry out to get a sense of like how many cells are there of both red cells and white cells. The differential count we carry out so that we can figure out what types of white cells are present and in what proportions. And then the chemistry panel, the protein electrophoresis, and the acute phase proteins are all tests that we carry out on the plasma, which you'll see in that photograph there. Those are spun down samples of blood that we've collected from an animal. And we've separated out the red cells, which are on the bottom, they're dark, and then the plasma, which is on top. So we can go to the next. So again, you know, the hematocrit really just gives us sort of like a very basic estimate of health. It'll tell us if an animal's anemic. Also, the white cells might form a really thick band. So you'll see in that picture, the red on that side is all of the red cells packed to one side. We can determine what % of the blood sample is red cells. The clearish section is the plasma. we tend to break that little tube and we'll put it on a refractometer, and that'll give us a sense of what the protein levels are in the animal. That gives us a quick snapshot of whether the animal's been eating recently, whether it's osmoregulating well, and other senses of its health status. And then there's a teeny tiny little band in between those two sections, and that's all the white cells. And sometimes that's really thick and can tell us whether or not the animal has inflammation. The other counts that we do, which I honestly don't do on the ship because it's very hard to use a microscope on the ship when we're not at dock, and it tends to make everybody seasick. We do carry out red blood cell counts, so we want to know how many red blood cells are present to determine that the animal's healthy. We want to know how many white blood cells are present. So I think, as you know, when you go to the doctor, if you have a white blood cell count, that means you're having an immune response. So that's something we're interested in determining, both what's normal in an animal versus whether animals are showing a response. And then platelet counts. Sharks don't have platelets. They have a cell type called a thrombocyte, but that's something that I thought people might be a little more familiar with. And then as far as the white blood cell types, it's important to look at what types are present and then the maturity of those cells in order to determine whether there's an immune response. So we can go to the next slide.
Real quick, Harley, because I don't want to leave some of our viewers hanging here, because there is at least one or two good questions, and you might get to this one in a sec, because I know it's a part of your study. But does taking these samples, Brittany asked, does taking these samples have a negative impact on the shark? And if you're going to get to that in a sec, because I know it's a part of your study, just so you can answer that in a sec, but I just want to make sure that we're not leaving our viewers hanging in terms of questions.
Sure. So actually taking the sample from the animal, we take a very small blood sample, so it doesn't impact the overall, health status of the animal. As far as any study that studies wildlife, in order to get these types of samples, you're interacting with those samples. So we always talk about the fact that what we're creating are kind of relative reference ranges. Obviously, there is that acute moment where you're handling the animal that's very different for it, and it can have impacts, but we try to take that into consideration. It's one of the reasons it's so important to have so many of us on the lift, is so that we're very fast, we get things very quickly, and if there's going to be any changes, that we catch those blood samples before those changes can happen. But overall, the blood volume that we take is very small, and we do calculate it out. Particularly when we were doing the young of the year sharks, we definitely were very critical of how much blood was appropriate to take from those animals because they were so small. So for white blood cells, I just wanted to provide you a picture of what white blood cells look like in a white shark. I also am showing you an erythrocyte, which is a red blood cell, and then a thrombocyte, which is the equivalent of a platelet in a white shark. The thing about sharks is we're still learning a lot about what their normal cells are, how they actually function, and we're often comparing them to how they function in comparison to mammals. And we do know that their differential count is very different from other species in that they have predominantly lymphocytes, whereas most other species tend to have more granulocytes, which are these neutrophils. And in shark species, we have these two special types of cells called fine eosinophilic granulocytes and then the coarse eosinophilic granulocytes. I imagine there could be physicians out there, so I'm just going to say that the coarse eosinophilic granulocyte, we do think functions like an eosinophil, and we think that the fine eosinophilic granulocyte functions as another phagocyte, sort of like a neutrophil. So we can continue. So chemistry panel, again, it's really just to give us a snapshot of organ function and metabolism. So we're just looking at organ health. So we're looking at liver function, kidney function, muscle metabolism, electrolyte balance. And again, as I said, we're trying to create this normal reference range for comparison. And as I've said in many talks before, we're trying to capture this now because we know that the environment's changing. We know that a lot of things are changing. And if we don't create these normals now, we're going to have nothing to compare to in the future to know that these disturbances have impacted the health of these animals. I did include an actual result from an animal. So this was the chemistry panel that we got back on white shark Hal. Just so you can see, we run a panel of 17 different analytes that give us kind of that snapshot picture of organ function and metabolism. So we can go to the next slide. So plasma proteins are super important in sharks. And the reason I'm going to tell you that is because if you deal with mammals or you deal with other species, we're very fortunate in the fact that we are kind enough that when we're sick, we mount a nice cellular response. So we'll have a nice high white blood cell count, and we'll give out clues early on that something is going on. We are finding that sharks don't do that. Sharks are very much like cats. They're very secretive and they tend to not have big responses. So we're trying to investigate different ways of determining whether or not these animals are responding to different stressors or other illnesses. And we do that by looking at the proteins that are in the plasma. And we do that in the primary way is by doing electrophoresis. So what you see on the side, it doesn't matter technique or anything, but what it's doing is it's separating out the proteins in the blood. There's well over 200 different kinds into different fractions. And by looking at the percentage of the protein that is that fraction, we can then start to get a sense of whether there's a disturbance in these proteins that might indicate that the animal has some sort of underlying inflammation. In addition to that, some of these proteins are very specific to mounting sort of an immediate kind of inflammatory response in response to infectious agents or other stressors. And these are acute phase proteins. And the one that I think most people are most familiar with is C-reactive protein because it's used really often in humans for a number of different reasons, but most often in cases of heart attacks or other sort of stressors like that. So it's something that I think a lot of people may have run, and we're finding that it actually is very useful in sharks. So again, this is an electrophoretic on your right. These are real results from real animals that that are on the tracker right now. So this was white shark yeti. This is an animal that we also caught in 2016 off of Nantucket. We can go to the next slide. So why is all of this important? Well, this is actual sort of preliminary findings that we have on the animals that we've studied to date. And when you look at sort of everything that we've been looking at together, white blood cell counts and different things, one of the things that has been shaking out is the fact that this one inflammatory protein that's elevated when animals are either stressed or having some sort of inflammatory process is that sharks in the southeast have a significantly higher level of CRP. And those are animals that we're typically sampling in the winter than animals that we catch in the northeast in the fall or animals that we catch in the mid-Atlantic in the summer. So that asks another big question. What is it about being in the southeast in the winter that is causing this type of response? It could be environmental. It could be social. We just don't know. But knowing they're having this response is a great place to start and ask questions. And that's why I have folks like Lisa Crawford and Lisa Hoops so that I can go to them and I say, well, what did you find in these animals? Is there anything that correlates between this high level and some sort of disturbance in what you're seeing? So we can go to the next slide.
Awesome. Thanks, Harley. I'm going to pick up from here and talk a little bit about my area of interest, which is nutrition and feeding ecology. And nutrition is a pretty important component of animal health because it decides whether you are healthy or not. It's the cornerstone. So having proper nutrition on board, especially for humans and cats and dogs, but specifically in fish, can help an animal overcome an infection, can help an animal with wound healing. If there are deficiencies in any particular mineral or vitamin, that causes disease down the road. So having nutrition right on the front end for an animal is vital to it staying healthy and becoming part of a viable population. So this is my particular area of expertise. Next slide, please. So similar to what Harley just talked about, we use blood to look at a variety of nutritional parameters in these animals so that we can also begin to establish baseline values for their nutritional health. And we're looking at a couple of things. We're looking at trace minerals and vitamins. Trace minerals are important because they perform a variety of functions in the body. And the key word there is trace. These are minerals that are required by the animal in very small quantities. There are 15 recognized essential trace minerals in fish, and we're looking at a small selection of them. But they play a variety of roles, ranging from becoming constituents of certain tissues. They're enzyme cofactors in making certain processes in the body work. They play a role in cellular, both intracellular and extracellular ionic balance, and they help regulate our endocrine systems. So they play a wide variety of roles. Now for fish, they can take their trace minerals in two forms. They can either get them from their diet, or they can actually absorb them across the gills from their water source, both freshwater and saltwater, depending on what fish species we're talking about. So some fish take them preferentially, their trace minerals preferentially from the diet, and others take them preferentially from the water. So it's important to try and understand these different routes and fates for these minerals, and how the animal actually balances them within their body. Some of these trace minerals are very tightly regulated, and some of them are not. So when we go ahead and run these panels and begin to look at some of these parameters, we can get a nice idea of how the animal is doing nutritionally based on where we see some of these levels. So from a trace mineral perspective, we're looking at cobalt, copper, iron, zinc, molybdenum, selenium, manganese, and iodine. If you're having a flashback to high school chemistry, yes, these are the same minerals that you would see on a periodic table. And again, they're required in very small amounts. And this is true of us as well. These are the same things that you would find in a multivitamin, some of these minerals as well. We do also require some of these things in very small quantities for our health. The other component that I'm interested in are vitamins. And I think most people are familiar with what vitamins are and what they do. They're essential for normal growth and reproduction, for nutritional health, and for actually regulating metabolism. There are 15 recognized essential vitamins for fish. And there's some of the same vitamins that we require in our diet. Most of them cannot be synthesized by the body, so the animal actually has to get them from the different things that they're eating. I'm particularly looking at, for this study, vitamin A and vitamin E, which play a variety of roles in eye health, membrane health. Vitamin E is an antioxidant and helps with regulating immune function and keeping oxidants down in the body. Next slide, please. The last component that I'm looking at for understanding baseline nutritional health in white sharks and other species of sharks is really looking at fatty acids. And fatty acids are simply the active components of lipids or fat. There are many different forms of fatty acids, and we can break them into classes. And this is probably what most people recognize when we talk about saturated or unsaturated fats. Those are basically groups of fatty acids that have very different chemical structures. And most people associate the saturated fats with kind of the unhealthy fats, and the unsaturated fats with the more healthy, or what we can refer to as omega fatty acids. So depending on their double bond structure, we won't go too deep down the chemistry hole at this point, but we can have omega-3 fatty acids, we can have omega-6 fatty acids, and we can even have omega-9 fatty acids. And they all play different roles in the animal in terms of their health. Fatty acids are really important for eicosanoid production, which is part of your inflammatory response in the body, or your anti-inflammatory response in the body, depending on which fatty acids that we're talking about. So the reason that a lot of folks take omega-3 fatty acids or fish oil is because they're great anti-inflammatory fatty acids, and they have a lot of benefit to the body. And the same is true for animals as well, cats and dogs, and even sharks. So when we go ahead and have our blood run for fatty acids, we get a nice profile of about 55 individual fatty acids and their proportion within that sample. And what's kind of interesting about fatty acids, besides providing a really nice marker of health and looking at health in the animal, is that they can be used as a biotracer of diet. It's kind of a secondary cool function of them. Specifically in fish, they are not able to create their own long chain polyunsaturated fatty acids. They have to get them from their diet. And so that means we can use a suite of those fatty acids to actually trace what they've been eating up the food chain. Next slide, please. And so this is a principle that's called fatty acid signature analysis. It's a really emerging technique in with sharks. It's been well done, documented in marine mammals and some other species, but it's based on the principle that you are what you eat. And so if you look at the food web here, you can see different things at the bottom of the food web. And they are able to incorporate different fatty acids within their tissues. And those fatty acids move up the food chain as different things eat them and stay relatively unmodified. So that when we get to the top of the food chain, we can actually take a tissue sample from our consumer and learn something about what they have been eating on, because these fatty acids are conserved across the food web. And so this is kind of an interesting and new technique related to sharks. It's often paired with stable isotope analysis, which is kind of a similar concept. The difference is that fatty acids, because we've got 55 of them to look at and kind of look at their combination, provides a much finer scale picture of dietary intake in these animals. So if we look at this figure, if a white shark was eating marine mammals, which are composed of blubber, we would expect to see this combination of fatty acids versus if they were eating something like a fish or cephalopods like squid. Next slide, please. And so I just wanted to show you a little bit of the data that we're starting to accumulate for the white sharks that we've been sampling to begin looking at diet in these guys. And so this is just a spatial representation of the fatty acid profile. So all 55 of those fatty acids and how they fit in sort of a spatial representation for the different age classes of animals that we've grouped them into. So what you should be able to see right away is that the young of the year, which are our green squares, have a very different diet composition than the triangles, the blue triangles and the orange triangles, which are our immature and our mature animals. So that's probably not really surprising, because as these guys grow and they get bigger and they move into different habitats, they probably have access to bigger and different things. Next slide, please. If we break this same data into capture location, we can start to see another interesting emerging pattern here. Again, the animals off to the left side, those red triangles are our young of the year, and everything off to the right side are our immature and mature animals, but now they're broken down by region. And so what you can really see is there is some conservative diet composition within each region. So in other words, the animals within Nova Scotia, those blue circles, are feeding on something similar but slightly different than, say, the animals in Nantucket, which are the diamonds at the bottom. And so this really begins to let us explore what types of foods they may be feeding on in these different regions. So if we lay the fatty acids, and that's what those lines are and those vectors, on top of this data, we can see the fatty acids that are driving the differences between these groups, and begin to look at those combinations of fatty acids and see if we can make some predictions about what types of foods these animals are feeding on. So we know that some of the animals in Nova Scotia are feeding on marine mammals. And that's because we see that signature of blubber showing up as some of the drivers of these differences. We also have some great evidence of that when we collect a fecal sample from these guys. So we have seen hair in the fecal samples of the animals that we have sampled in Nova Scotia. There's a big seal population up there, so it makes sense that that's what they're feeding on. So it is some of these clues and the beginnings to untangle all of these combinations of fatty acids that we're going to be able to hopefully dial down a little bit and get some specific information about their diet. And this is really important for not only looking at baseline nutritional health, similar to what we're trying to establish with all of the great blood data that Harley's been collecting, but we also want to know how these animals are using their habitat. So when they're coming into these different areas and they're using them at different times of the year, why? Are they feeding on something different? Are they coming in to reproduce? Can we distinguish any of that from some of this data? And then finally, the last thing that's really important is to try and understand how these animals may be impacted going forward by climate change or any anthropogenic changes that may impact them. So, you know, if they move into areas that are heavily impacted by contamination, does that change their diet? Does it make them does it make certain prey items less available to them? So that is kind of where we're trying to go with this data, and stay tuned as we collect more animals and we do a little bit more fancy statistics. We'll hope to see if we can tease some of this diet information out a little bit finer.
Right, and that brings us, that's a great segue into what I work on, which is ecotoxicology, and talking about feeding ecology and nutrition and the changing environmental factors that may affect diet and feeding ability is really highly related to toxicology, because ecotoxicology is just the meshing of ecology, so animals in their environment and how they interact with their environment, and then the larger study of toxicology, which is just the study of toxins and poisons and the effects they have on the body. And so when I'm studying ecotoxicology of white sharks, I'm really looking for the effects of toxic chemicals that these white sharks are getting from their diet at the individual level, the population level, and then at that larger ecosystem level, so I can get an idea of health at all these levels of organization. Now, white sharks in particular are highly susceptible to organic contaminants and toxins because they're apex predators. They're feeding at the top of the food chain on prey items that tend to have a lot of contaminants. White sharks are also very long-lived, so as they grow older, grow bigger, and are eating more, they're accumulating more toxins in their system over time. And then something that might be interesting to viewers at home is that sharks, all sharks, have extremely large fatty livers, and organic molecules and organic contaminants actually preferentially absorb into the liver. And so sharks with their big livers can be more susceptible to accumulation of all these toxins. And this can kind of have a poor outcome for sharks because they tend to grow slowly, they mature slowly, and they have low fecundity, meaning they don't have very many babies and they don't have them very often. So if our sharks are having these negative impacts of contaminant exposure, there's the potential that their populations might be affected and they might not be able to rebound because of these life history characteristics, growing slowly, maturing slowly, and not having a lot of babies to replace any that are lost. Now, these are just the molecular representations of the toxic chemicals that I study. So in the top left, we have PCBs, which are found in industrial processes, electrical transformers, things like that. They're not used today, but they are legacy compounds in the environment. Then the top right, we have pesticides. I'm looking at a whole suite of pesticides, even ones that you might use in your backyard on your lawn, but also pesticides that are used in more large-scale agricultural processes. And then on the bottom left, that's PFAS. And PFAS is a chemical contaminant that's found in nonstick coatings or firefighting foams. This is an emerging contaminant that we don't actually see in high concentrations yet in the wild. However, because of the prevalence of these types of nonstick surfaces and firefighting materials, we are starting to see PFAS end up in drinking water, groundwater, and it is making its way into the ocean. And we have a theory that PFAS might be ending up in our top predators like the white sharks. And then finally, in the bottom right there, we have methyl mercury, which is just the organic form of mercury. And I'm sure many of you are familiar with mercury in seafood in particular. And again, mercury is a natural occurring element, but a lot of mercury is released into the environment through human processes. And if there's one concept I want to leave you with for this section, it's that these contaminants are highly persistent in the environment. Once they get there, they're very hard to remove and they don't easily break down. And they're also very bioaccumulative. So they're going to end up in the food chain, and they're going to concentrate in high values in our top predators. And finally, they are extremely toxic, and they may be posing adverse health effects on wildlife. And so how do these contaminants end up in the ocean? Well, if we're looking at our persistent organic pollutants, those things like PCBs, PFAS, those tend to come from industrial processes, large-scale agriculture. They can be directly run off into the water or leached into the groundwater. There's also things like oil spills that can be natural or come from disasters or accidents. And then a lot of the toxic metals, such as mercury, come from large-scale industrial processes, particularly coal burning. And those contaminants can enter the waterway through runoff, direct dumping, or even through atmospheric deposition. When we burn coal, mercury ends up in the air, it's transported across the ocean, and then is rained down into the ocean. And so what I want you to understand here is that all of these chemicals that I'm looking at come from humans. They are anthropogenically derived from human processes, and they're ending up in the oceans, ending up in our sharks, and we need to now investigate the potential health impacts of these contaminants on our sharks. And so because most people are probably familiar with mercury, I wanted to show you some of my preliminary mercury data that I have. So to orient you to this graph, on the x-axis on the bottom, we have precaudal length. That's the length of the shark from the tip of the snout to the base of the tail. And then on the vertical y-axis, we have the total amount of mercury in muscle tissue. And what you can see is that as the animals get larger, they tend to have more mercury in their tissues. And that's what we would expect, right? As these animals are growing older, larger, and eating more, they're going to be eating more contaminated prey and accumulating mercury in their tissues that I can then measure when I go to take a muscle sample from our white sharks on the lift. And so I've added in the names of some of the sharks you might be familiar with. We've got caper down at the very bottom left with a very small mercury value, which makes sense because caper was a pretty small animal. And then if you look all the way up at the top, we have Hal with an absolutely out of control mercury level, very high, but still what we would expect. This was a very large animal. We would expect to see Hal having a lot of mercury in his tissues. And one of the things I want to point out here is if you look at the blue dots from the Southeast US, or what we call the NASFA region, if you look at Helena and Caroline, those two sharks were about the same size. However, Helena has much more mercury in her muscle tissue compared to Caroline. And so that's something that kind of piqued my interest of why do these animals, both females from the same region of the same size, why would they have such drastically different mercury levels? And so that's something that I took to both Harley and Lisa to ask them, did you see anything anomalous in your data that might indicate why we would see this huge discrepancy? And so Lisa can look at the fatty acid profiles and the nutrition to make sure both animals were eating and to see what they were eating. And then Harley can look at the immune response in those blood panels to see, was Caroline maybe experiencing an immune response that would indicate something about her feeding or potentially her mercury ingestion. So while the data gives us a lot of answers to our questions, it also allows us to pose a lot more and gives us a lot more to investigate. And just in case these mercury values don't really relate to anything in your mind, I want to leave you with some kind of tangible example. So Hal, with a mercury concentration of about 25 milligrams per kilogram, that's very high. If that concentration of mercury were in an animal that we would eat, like a tuna, and you were to go and have a sushi dinner of that tuna, you would be consuming 110 times the safe weekly limit of mercury as set by the EPA. So that gives you just an idea of how much these contaminants can bioaccumulate in our white sharks and the potential impacts not only on their health, but human health and ecosystem health as well. And so then to just tie everything together, we've touched on health within an animal, looking at its diet and nutritional status, and then health of the environment as well. We have these chemical contaminants that enter the environment. We have other anthropogenically derived pollutants such as plastics, and these all intersect to give us an idea of animal health. And so we've set up these investigations to help us answer our larger questions about how is animal health being affected, and how can we start to tease out what is normal animal health versus what is abnormal animal health that requires further investigation.
Great, that was wonderful, Lisa. So some of the ways that we start interacting with each other, these veterinary health panels and contaminants. So what we're looking for are relationships between the contaminants that are present in the animals and the levels and blood biomarkers. So we want to see if they have these contaminants, particularly new ones that we don't know a lot about. We want to know whether or not they're really impacting the animal. Is their liver function the same? Is their kidney function the same? Are they able to metabolize the same way? Are they responding the same way that we would expect when we're handling them? So all those things are important to answer. We're also trying to link those contaminants with those stress response markers. So a lot of those protein evaluations that I showed you are really giving us a sense of, if these animals are exposed to these contaminants, if they have high levels or low levels, is there a difference in those protein levels? And the same thing for immune response. We're trying to see, are they having a high white blood cell count? Do they have higher than normal ratios of granulocytes to lymphocytes? The kind of things that we look for just to see, are these contaminants actually impacting or changing the health of the animals? And as I've said before, and we'll say again, it's important to ask these questions now, because the reality is this is all going to change in 50 years. So we need to see how everything is shaping up now so that we can look at things in the future.
And along those same lines, we definitely have a link, and hopefully we've done a great job of showcasing that to you, between nutritional status in animals and their contaminant loads. So as I mentioned, a lot of the minerals that I'm looking at are required by these animals in very small amounts. However, if they get exposed to them in very large amounts, they can become toxic and are often referred to as heavy metals at that point. And so through environmental exposure, these animals may have trouble regulating their mineral balance when they are exposed to too much of a certain thing, either through their diet or just through environmental exposure. But there is another interesting link between some of the nutritional data and the contaminant data. There are certain minerals and vitamins that help an animal fight off the effects of some contaminants. And particularly, we can look at the trace mineral selenium, which works in conjunction with vitamin E in the body as an antioxidant and helps protect the body from some of these environmental assaults. And when animals are exposed to mercury, selenium can actually attach to the mercury molecule, making it less toxic or having less toxic effect to the actual animal. So something that Lisa and I will begin to continue to explore is the relationship between selenium as the animal picks it up from their diet, and the role or the volume of mercury in their tissues as well. And that helps us hopefully to understand how the animals are handling some of this environmental exposure. There are certain vitamins that also can help combat the harmful effects of some of these contaminants as well. So there's a really nice connection between diet and contaminant exposure, which ultimately provides the information that we need to understand animal health. So hopefully we've done a really good job of explaining to you and sort of showcasing how each of us have our individual areas of interest, but ultimately we can't get a picture of the animal without working together to understand all of the pieces to the puzzle. So I'll let Lisa Crawford jump in for our last slide.
So with that, we've told you all about our research, and, not only are we working together to answer some large questions with our individual studies, all of our work intersects, and I'm so grateful to be able to work with these wonderful researchers all together on the lift. And you can see pictures here of all of us collecting our samples from white sharks on the lift. And then to leave you with one final Venn diagram, since we've shown you quite a few, one more piece of information you need to know is Harley's real name is Elisa. And so we have the Venn diagram of Lisa's. And we all overlap not only in name, but also in our love and appreciation and desire to help sharks and protect our oceans. So with that, we would thank you, and we'll take your questions.
That was awesome, guys. That was some real science right there at the end. Well done. That one quick question I have here before I have a question of my own. Patricia says her 7-year-old daughter wants to know, and you kind of touched on this. How do you test for contaminants in sharks? We said, Crawford, do you want to take that one for Patricia?
Yeah, so primarily for my research, I collect a very small muscle sample, but I can also use blood. And what we do is we dry these samples, we freeze dry them, and then there are actual machines that are designed to burn the samples and then detect how much of each individual element or contaminant is in that combusted material. So it's very nice that these machines are available to use, because you can put a sample in, hit go, and have it run. Some of the more complex organics, like the PCBs and PFAS, those require more complicated chemical techniques. And so we usually collaborate with chemists to help us extract those chemicals from the tissues and then put them in the machines to actually quantify the levels of contaminants. But great question from your 7-year-old. That's awesome.
Yeah, so to make it a little bit more simple for the 7-year-old, you get the sample from the shark, then you take it, whether it's the blood sample, the muscle, tissue, whatever, and then you take it back to the lab, and then you look using science equipment. If anybody else has any questions, now is the time to leave them, and we'll jump to them. I do have a quick question. So really, this is we're basically helping to develop our most comprehensive understanding yet of white sharks. It's kind of a 300 and sort of a, not quite 360 degree view, but why is getting this knowledge so important in terms of helping to protect these animals moving forward? I don't know who wants to take that one, because it made it very clear that you guys are all working together to put this awesome picture together. Now I'm just curious, with this really comprehensive view, what can we do to help protect the sharks moving forward and our oceans? Don't all jump at once.
I can jump. I mean, as I said at the beginning, one of the critical things is really just understanding population level health and using these white sharks as a sentinel for our oceans, finding ways to detect disturbances either regionally or seasonally, life histories that are under pressure, and then using that to develop smart policies so that we can protect animals in certain regions if they need it, at certain life stages if they need it. And ultimately, that's the goal of most research, is really to be able to contribute to informed policy, science-based policy. So we're making sort of the right decisions for populations and our oceans based on the knowledge that we get from these studies.
Okay, another question here from Bill. Can we get, Lisa Hoops covering up. Can we get an age estimate other than through recapture, possibly higher mercury and older sharks that are of similar length? Or I guess the question is there a way of being able to age sharks? And I will put your face back up there, Lisa.
So it's difficult to age a shark while it's still alive. But we can use the estimate of its body size and body weight to tell us an approximate age based on other studies. So what I can do for each shark that we catch, knowing its length and weight, I can go and look at some tables of associated ages and body sizes to tell me approximately how old that shark would be. And then, yeah, you're correct in saying that as these animals grow bigger and older, their mercury levels should be increasing. The dream would be to recapture one of these animals, be able to resample its mercury and other contaminants multiple times. But yeah, I can get kind of an estimate of how old the shark is in relation to its mercury level. I hope that answers your question, Bill.
And I'm just going to jump in really quickly and tell you that life history information in sharks is something that's incredibly valuable and super hard to get. A lot of these age estimates are based on sort of vertebral body analysis. And it's something that a lot of people are working on, and it's very species specific as well. So, you know, lengths in a white shark don't necessarily equate to other species. So it's really critical that there's other researchers out there that are doing this work, because we use that as the basis of a lot of work that we do.
There's a good question here. It just jumped. Are there other researchers / partners contributing white shark samples to these particular studies besides what sharks are being caught by OCEARCH?
I mean, I can answer that. I do work with other partners. I work with partners at NOAA and the South Fork Natural History Museum. That Young of the Year white shark study that we started in 2016 and 2017. We've been continuing to do some of that study, both trying to get a little better sense of their movement, their habitat use, and I've been continuing a lot of those health studies in those animals as well. So yes, I mean, all of us, we're partners with OCEARCH, we're partners with each other, and then we have other partners outside of that. And I think that's what makes some of these studies so strong, is just having those kind of partnerships and samples for other places.
And as we start butting up against our time here, we'll take just a couple more quick questions here. I think this one, Lisa, will be for you. I'll put it up over your face again. Is there a use for shark nutrition information for humans or for other sea life? Using what you, I guess the question, using what you do, can we take this and sort of apply it to how it might have an impact on us as humans?
Yeah, absolutely. I think, understanding what these animals are feeding in the wild and how they're using their habitat helps with their own conservation, but helps us to understand how we're using some of those same resources as well that the sharks may be relying on. Looking at nutrition specifically, as a nutritionist at an aquarium, everything that we learn from wild sharks is applied to the sharks in our collection. And as apex predators, while we don't have white sharks in our collection, they're feeding at the top of the food chain. And so some of their fatty acids and their mineral and vitamin compositions help me manage the diets for some other species that are also at the top of the food chain. And vice versa. What we learn about the diet and the feeding of many of the sharks that we have at the aquarium, we can also take that information to help us learn more about some of their wild counterparts. So I think this exchange of information goes both ways. And certainly, the more that we learn about the ocean, the more we learn about ourselves. So as the ocean goes, so does everything else, including human health, right? That's that One Health tie from the beginning of this talk. So understanding the health of our oceans and the inhabitants in it really provides us a mirror into our own health as well.
And correct me if I'm wrong, Lisa and Lisa. Also a direct impact on humans is what sharks are eating. Sometimes us as humans eat the same thing, right? So if we're, say, through Lisa Crawford's study, seeing that, some of these toxins are building up in sharks, there is the potential that we are eating some of those same toxins too. Am I correct, Lisa?
That's exactly right. White sharks eat tuna. They eat other big fish, billfishes that we would eat as well. And so if white sharks are getting these contaminants from their diet, and we tend to eat the same diet, we are also then accumulating those toxins. Now, of course, we eat a lot less than a white shark, but it's important to make, safe seafood choices, understanding that there are some fish with higher mercury and higher contaminant levels. And so it's a good idea to diversify your seafood eating.
Cool. We have time for about 2 more questions here. Harley, I think this one will be for you. Do white sharks have different blood types?
I get this question a lot about sharks, and I'm going to answer it just generally for sharks. I will tell you as a veterinarian that works with sharks that this is a question we have a lot. There have been some very limited studies where we've done kind of rudimentary cross-matching. So basically separating out the red cells and then intermingling plasma. We know that within the same species of shark, often it's safe to do a transfusion. That would suggest that there's probably not separate blood types, but again, this is very rudimentary studies. There also have been instances where we have transfused from different species that are related and not had any reactions. So, at this point, what determines a blood type is pretty complex, and it has a lot to do with what's on the surface of the red cells. So I'd say some of the preliminary work would suggest that they probably don't have different blood types necessarily, but that's just a best guess based on what we can do right now.
Excellent. And then for our last question here, Patricia has a question from her 11-year-old daughter. She wants to know, what is the most common element found in the samples?
So at least from a contaminant standpoint, and this is a really good question. Patricia, your kids are asking great questions. It's not necessarily that certain elements are more common than others. We go looking for certain contaminants. So I have an idea in mind. I want to look for mercury, PCBs, pesticides, and PFAS when I go looking into this tissue. And then depending on the identity of the contaminant, different concentrations in the tissues may be differentially toxic across species, body sizes, that sort of thing. So when I'm looking for mercury, I'm looking for concentrations on a certain scale that would tell me, oh yes, this animal's mercury value is very high compared to another animal. If I'm looking at PCBs, because they're not still today put into the environment, we see much lower levels of PCBs. However, the toxic effects of PCBs at a very low level might be more extreme than mercury at a high level. And so that's kind of how I have to look at my study, is not only what is in the tissue, but at what levels, and what levels might be having a negative health effect on the animal. But yeah, thank you, Patricia, for your wonderful questions.
And we are butting up kind of against our time here. But I do want to thank everybody who joined us. I saw people chiming in from basically all over the world, from all over the US and even the Philippines and beyond. So it's so great to have so many of you here contributing, participating. It means a lot to us. So thank you. Also, thank you to all of our rock star collaborating scientists who showed up today. Lisa, Harley, you guys were awesome. Like I said at the beginning, I had a hunch that even myself, I might learn at least something, and I definitely learned multiple things. And that seems to be a trend that anytime I talk to either one of you. So thank you so much for bringing your expertise here and sharing them with us all. Does anybody have any last thoughts before we sign off here?
For me, it's just thank you for everybody for watching and being interested in all the research that we do. Thank you to the 2 Lisas for working with me, and thank you to OCEARCH to giving us access to all of these amazing samples and animals.
I couldn't have said it better myself. Yes, thanks to everyone for watching. I saw some friends and familiar names along the chat. So thanks for your support. And we're always looking forward to the next expedition where we can get our hands on some more sharks and continue to try and answer some of these really important questions. And it is just a delight to work with these fabulous scientists. So thanks, OCEARCH, for bringing us all together.
Yep.
Thank you, everyone. Yeah, thank you so much, everyone, for tuning in, asking really awesome questions. Hopefully, as we move forward with our research, we'll start to answer some of these big questions, and then we'll get to update you as we get our results coming in. So stay tuned. Please stay interested in sharks and interested in the work of OCEARCH, and hopefully we'll come back to you soon with more.
Yeah, we're all looking forward to it. Thanks again, everybody. And I think with that, we'll sign off.